have a query to you. Actually, yesterday, actually, I couldn't present my poster. The poster number was P104. That means P104. But when I
Okay.
email the organizing committee, they uh, mailed me that actually I can present my poster uh, at 15 GMT. 15 GMT.
Okay.
That means after 16 minutes. According to the actually, may I present my you can say thesis or work today or right now, or I should wait for the
So,
next session?
Professor Priyani, shall we allow
It
him?
should
Yeah,
be,
I
it
think.
should be.
Yeah,
Yeah. Let
yeah,
let
yeah.
him.
You can present. Yeah, you can start. Yeah.
Okay, thank you, sir. Sir, are you able to, sir? Are you able to see my screen?
See your slide and yeah, hear you.
Okay, thank you, sir. Thank you, everyone. And I'd like to present my work that I have done in my master's thesis. And the work was basically synthesis, antimicrobial evaluation, docking and dynamic investigation of novel steroid monoperoxide. That was my, uh, you can say, Commonwealth Chemistry Posters ID. At the same time, poster ID was P104. And right now, I'd like to present my overall tasks that I have done. Actually, our objectives was basically, at first, I'd like to summarize the overall things. At first, we have chosen some compounds, the compound that have some, you can say, antimicrobial properties, and we have chosen methyl alpha D manoperoxide. That was our starting material, and in our chemistry laboratory we have chosen this compound actually by studying different kinds of you can say previous articles or scientific articles, and we have seen that actually methyl alpha D manoperoxide that have awesome you can say antimicrobial properties, and if we change the free hydroxyl group from this compound, we will be able to get some exclusive antimicrobial properties of our synthesis products. And with the view to actually we have done lots of things. That means at first we have run, you can say, experimental tasks. That is our starting material. And uh, actually with the help of some, you can say, pyridine and acylating agent, we have completed reaction. And in this reaction, to make the faster, we have used actually a catalyst, that means DMAP, dimethyl aminopyridine, for making the reaction faster. And similarly, when we have completed the first reaction, we have seen that actually that is the, you can say, six number free hydroxyl group. When we have completed, you can say, sterilization, then six number free hydroxyl group is blocked by the sterile you can say acylating agent. And that is our first product. And from this product we have further done acylation reaction using different kinds of acylating agent like octanoyl chloride. And the number three compound is acylating agent is octanoyl chloride, and number four is, you can say, decanoyl chloride, and number five compound is, you can say, dodecanoyl chloride, and that is a, you can say, hexadecanoyl chloride acylating agent. So we have we when we have changed the acylating agent, we have found different percentage of product, and the percentage was, you can say, 81%, 76%, 83%, and 66%. That means compound 5 was, you can say, huge amount. The percentage was really high. That was 83%. Then when we have completed you can say this acylation reaction, we have found, you can say, 2, 3, 4, 5, and 6 number products. And we have done some computational calculation. That means we have draw the synthesized product structure using, you can say, Gaussian software. And then we have we have using smiles, and we have run, you can say, pass studies. Pass means probability of activity spectra of substance. That means our synthesis products have, you can say, specific activity or not. That means it have any antibacterial, antifungal, or anticarcinogenic property or not. That we have done. So I would like to show you that actually when we have done pass analysis, that means probability of activity spectrum of substance, then we have seen that our compound 3 that show the you can say highest antibacterial and antifungal properties. And at the same time, I would like to move in the next session, next you can say slide. That will be easier for understand because my poster is you can say uh, compressed kind of. Then when we have done you can say pass analysis, we have found that compound 3 that compound 3, 4, 5, and 6 that shows the highest antifungal, sorry, antibacterial properties. And that is you can say comparable to the tetracycline is a, you can say standard antibiotic you can say. At the same time, if you'd like to say that PA and PI means probability to be active, PI means probability to be inactive. That means we are seeing that the probability to be active is higher in case of compound 3, 4, and 5, and 6 in comparison to the probability to be inactive. And at the same time, we have found that actually compound 3 that show the you can say similarly antifungal properties that we have done from past analysis. And we have compared with some standard drugs like FCZ, that is called the you can say fluconazole, ICZ, that is called the you can say itraconazole, NOIS, that is called the nystatin, and TTC, that indicates the you can say tetracycline. And the next one, when we have done good antibacterial and antifungal properties, we have done you can say some experimental tasks on the basis of past studies. When we have get a good score from past studies, we have completed some experimental studies using microbiology department or microbiology lab from our you can say university. And we have seen that bacillus cereus, there is a bacteria, escherichia coli, there is another bacteria, pseudomonas aeruginosa, and klebsiella pneumoniae. uh, these are the four bacteria that we have done you can say these studies for you can say to check it out its antibacterial properties. And we used you can say Diggs diffusion method, and we have found that means our standard compound tetracycline that shows the 22.33 you can say inhibition zone, that was its 100 microgram deciliter in Diggs or per Diggs. And on the other hand, we are seeing that our compound 3 that show you can say good or comparable score that is you can say comparable to the tetracycline. At the same time, it show the you can say good score in case of you can say Escherichia coli. At the same time, that is not the fully equivalent or you can say comparable to the you can say standard drug, but from our you can say experimental drugs that we can assume that actually compound 3 that can show the good you can say antibacterial properties. And at the same time, we have done some antifungal properties using you can say this you can say fungal strains like Fusarium equiseri, Aspergillus flavus, and Aspergillus niger, and penicillin. And then we have seen that our fluconazole compound, that is a you can say antifungal you can say drug that is used in you can say our pharmaceutical industry in our country. And there is a good you can say pharmaceutical drug, standard drug. But our compound 3 that show the 63.89 antifungal drug, that means score inhibition score. So we can say that our compound, that is a uh, good antifungal drug that can replace the fluconazole. Those are you can say available in the market or you can say pharmaceutical industry. So we are assume that actually our compound 3 that can replace the position of fluconazole if it is go through the you can say in the clinical trials. That is our assumptions from the you can say computational studies, sorry, experimental studies. When we have get the you can say good antibacterial and antifungal you can say studies score or inhibition zones, then we have done some computational tasks that is called the, you can say molecular docking. From the molecular docking, we are able to check it out. That means our antifungal protein, that means we know that about each and every antifungal protein that has you can say specific protein ID. That means we know that about 3LD6, that is a protein ID of you can say uh, fluconazole or Candida albicans. That is a fungus. Actually, when we do molecular docking using our synthesized drug compound
Has,
against,
Hasinullah, can you please summarize your talk?
okay,
Sorry to interrupt you. Can you please summarize it? Yeah.
okay, I'd like to see. Okay, we have done molecular docking and we have done molecular dynamic simulation to check it out. That means uh, our compound that can inhibit the that means fungal protein or not. And we have see that actually our compound that has, you can say, good, you can say, binding affinity, -7.1. Fluconazole that show the -9.3. Our compound that show the, you can say, -7.9. Our experimental study and computational study that, you can say, corroboratively, you can say that actually this compound 3 that can take place, the position of the fluconazole in, you can say, medicinal sector, or you can say pharmaceutical sector. That is our outcomes. And in these specific studies we have done, that means we have synthesized our compound. At the same time we have checked out the biological activity, that means it is antibacterially active or antifungal active. We have seen that actually our compound 3 that show that, you can say, more antifungal activity. And at the same time we have checked out the computational, you can say, toxicity determination using protox 3 server. And we have seen that our compound that has, you can say, less toxicity. That means if that at first, I mean, I have shown that actually fluconazole that has, you can say, some side effects and toxicity and hepatotoxicity, but our compound 3 that show that, you can say, lowest toxicity, we have confirmed from our, you can say, computational study using protox 3. So that's all about my presentation. So if you have any question, then you can ask me.
Uh, Visitor, you can stop sharing your screen.
Okay, thank you, sir.